Hello everyone. Today we will talk about the specific immune response. We have already explained in a previous video the mechanism of the non-specific immune response, and we said that when the non-specific immune response fails to eliminate the intruder, the specific immune response takes place. Innate versus adaptive immunity. The innate or non-specific immunity is considered as the first line of defense against intruders, whereas the adaptive or specific immune response corresponds to the second line of body defense. The innate non-specific immune response is immediate. It is triggered a few hours later or few hours following the invasion. The adaptive specific immune response is delayed and long-lasting. It appears within a few days following the persistence of the infection. Now, the effectors of the innate immunity are mainly macrophages and neutrophils. So the major process is inflammation and phagocytosis, whereas the effector cells of the adaptive immune response are B and T lymphocytes. Now, for the antigen dependency, the innate immune response is independent from the identity of the antigen, whereas the specific immune response depends on the identity of the intruder. Here we have examples on the manifestation or the components of the innate immune response, which are the natural barriers, skin, hair, mucosal membranes, and the effectors are phagocytes, granulocytes. Here we have examples. We have also swelling, pus, pain, and redness, but also we have the response of T and B lymphocytes. As you can see here, we have the main cells involved in the rapid innate immunity and the main cells involved in the adaptive immune response, which is a slow and delayed response. The cells involved in the specific immune response are mainly B cells secreting antibodies and T cells, which are T4 or T helper cells secreting interleukins that will activate other immune cells. We also have the T8 cells or cytotoxic T lymphocytes that will lyse the modified cells of the modified self. Here we have an interaction between the innate and the adaptive immunity. This interaction will be discussed in other videos. So interaction is mainly between the macrophages that have digest the bacteria or the intruder and the T helper. So the macrophage digesting the bacteria will present the bacterial peptide in MHC class II molecules. And this complex MHC class II molecules plus bacterial antigen will be recognized by a specific T4 cell, which will lead to the activation of the T4 cell. In turn, will activate either B lymphocytes or T lymphocytes. All this mechanism will be discussed later. In our session, we will talk about the induction of the adaptive or the specific immunity. This immunity is referred to as acquired immunity or specific immunity. It is only found in vertebrates. It is specific to the pathogen presented, It is a delayed response since it needs several days to develop, and the effectors of this response are B and T lymphocytes. These experimental studies have shown that the immune response is specific. Vibrio cholera and Salmonella are two types of bacteria that cause serious intestinal infections. In this experiment, we have used two bacteria that cause intestinal infections. And these two bacteria were injected to chicken, and the results appear here. The Vibrio cholera and Salmonella are fatal. They cause the death of the animals. So, we submit these two bacteria to high temperatures so that the bacteria die, but their antigens are preserved. This is called killed bacteria. When the bacteria is submitted to high temperatures, it dies, but it can still trigger an immune response. The question is, justify, based on document A, is that the immune response is specific. Take a few minutes to answer this question. Why is the immune response specific according to document A? Since the injection of killed Salmonella leads to a subsequent resistance against live Salmonella, but not against live Vibrio cholera, as shown in the experiment 1 and 3, which means that the chicken injected with killed salmonella is considered as a vaccine, triggering an immune response and an immune memory. All these processes will be explained later. Now, the chicken survives following the injection of killed salmonella and the live salmonella 10 days later because the chicken becomes resistant to the live salmonella. It acquires an immunity against the harmful bacteria. Whereas if the chicken was injected with the live Vibrio cholera, after the injection of the killed salmonella, this chicken dies. This means that the killed salmonella gives an immunity to the chicken against the same bacterium and not against Vibrio cholera. Similarly, the killed Vibrio cholera gives a resistance or an immunity to the chicken against the live Vibrio cholera and not against the live Salmonella. So based on these results, we can say that the immune response is specific. Since the killed Salmonella gives an immunity against the live Salmonella and the killed Vibrio cholera gives an immunity against the live cholera. So based on document A, we can say that the immune response is specific. Now passing to the second series of experiment. This series of experiment will show us the types of the specific immune response. The specific immune response develops usually within 1 to 2 weeks following the contact with a foreign body. In order to understand what type of specific immune response is induced against a given antigen, the following immunity transfer experiments are performed. These experiments consist of inducing a specific immune response against a microbe or a toxin. Toxin is a molecule secreted by bacteria in one animal. So we induce a specific immune response in an animal, and then we transfer the different components of that animal's blood to another animal, and we notice, or we see the results. The recipient subsequently receives an injection of that microbe or toxin that is normally fatal. So first we vaccinate the animal, then we inject it with the harmful microbe. And according to the outcome, we can determine which blood component, the serum, the antibodies, or the T lymphocytes, provides the protection to the animal. Since we have already explained that the adaptive immunity, the effectors of this adaptive immunity are B lymphocytes producing antibodies and T lymphocytes. So, based on document B, we will determine which blood component, the B or T lymphocytes, will provide protection to the animal against the injected microbe. So in experiment B, in experiment A of document B, we study the immune response against tetanus toxin. This toxin is secreted by a bacterium called Clostridium tetani. This toxin is responsible for tetanus disease, which can be fatal. Now, laboratory manipulations can eliminate the harmful effects of the toxin without altering its antigenic determinants or epitopes. So we can, in the lab, neutralize the toxin. Yani we can eliminate its harmful antigens and preserve its non-harmful epitopes. In this case, the toxin is called toxoid. Toxoid means attenuated or neutralized toxin. Attenuated is a toxin deprived from its harmful antigens, but still capable of inducing an immune response. So in experiment A, we studied the case of a soluble antigen injected into a mouse A1. The question is, based on document B, deduce the effector cells of the immune response triggered against tetanus. Having a mouse A1 injected with tetanus toxoid, the serum of this mouse is extracted and divided into two parts, serum and blood cells. The serum was injected to a mouse A2 having received the tetanus toxin, which is harmful. But the mouse survives. It doesn't die. However, the injection of the blood cells deriving from mouse A1 receiving tetanus toxoid, these cells injected to mouse A3 having tetanus toxin injection dies. This indicates what? This indicates that the cells of the blood, mainly the T cells, are not involved in the immunity against tetanus toxin, which is a soluble antigen. However, the filtration of the serum of rabbit A1, then the injection of the filtrate to a fourth mouse A4 having received tetanus toxin leads to the death of the mouse A4. This shows that the effectors of the immune response against tetanus toxoid and thus against tetanus toxin are found in the serum and not in the blood cells. And this shows also that the serum of the immunized mouse A1 contains molecules that fix the antigen, since we see here that tetanus toxoid is attached to a solid matrix, so the filtrate doesn't contain this tetanus toxoid, so the filtrate contains nothing. Injected to mouse A4, the mouse dies. Thus, there is a molecule, or there are molecules found in the immunized serum that protects the mouse A2 from tetanus toxin. We can say that the serum contains antibodies, anti-tetanus toxoid antibodies. So, according to experiment A1, we can say that the effectors of the immune response against tetanus are the antibodies found in the serum. Passing to experiment B. In experiment B, we study the immune response against the Koch bacillus, which is responsible for tuberculosis. Koch bacillus is an intracellular bacterium. Like the virus, it enters the cell, causing infections in the lungs and other organs that might lead to death. The attenuated bacteria for Koch bacillus is Calmette and Guerin bacillus. It's an attenuated bacterium that shares common antigenic determinants with Koch bacillus. So, a mouse B1 was injected with CGB. Its serum is divided into two parts. Its blood is divided into two parts. Sorry. The serum injected to a mouse B3 receiving the Koch bacillus, the harmful intracellular bacterium, the mouse B3 dies. Now, the cells extracted from the blood of the immunized mouse B1 injected to a mouse B2 receiving Koch bacillus, the mouse B2 survives. This indicates what? This indicates that the effectors of the immune response against tuberculosis are the blood cells and not the serum. Now, these blood cells are divided into B lymphocytes and T lymphocytes. The B lymphocytes were injected to mouse before receiving the Koch bacillus, the harmful bacterium. This mouse dies. Whereas the T cells are injected to a mouse B5 also receiving the Koch bacillus, this mouse survives. So we can say that the immune response triggered against the Koch bacillus that is similar to a virus are the T lymphocytes and not the B lymphocytes. So we have seen two types of specific immune responses. An immune response whose effectors are antibodies found in the serum, and an immune response whose effectors are the T lymphocytes, mainly the cytotoxic T lymphocytes. Name, based on this document, the type of immune responses triggered against tetanus and tuberculosis. When the immune response triggers against soluble antigens or any non-self intruder or antigen. When the effectors of such as immune response are antibodies found in the serum secreted by plasmocytes, the immune response is called specific humoral immune response. And now, when the effectors of the immune response are T lymphocytes, mainly cytotoxic T lymphocytes. In this case, the immune response is a cell-mediated specific immune response. So we can say that there are two main mechanisms of the adaptive immune response, the humoral specific immune response and the cell-mediated one. The humoral specific immunity is also called antibody-mediated immunity, since the effectors of this immunity are antibodies. Now, with the assistance or the cooperation between the T helper and the B lymphocytes, these latter will differentiate into plasma cells secreting antibodies against specific antigen. So we talk about a cellular cooperation between T helper and B lymphocytes. B lymphocytes will differentiate into plasma cells that will secrete circulating antibodies against specific antigens. The humoral immunity deals with antigens from pathogens that are freely circulating or outside the infected cells, which means that once the intruder enters the cell, once the virus, for example, enters a cell, the antibody cannot recognize the virus when it is inside the cell. And thus, the antibody cannot bind to the virus or neutralize it if the virus is in the cell. The antibodies produced by the plasma cells bind to the epitopes of the antigens, neutralize them, and cause phagocytosis that will be discussed later. Now, for the cell-mediated specific immunity, it occurs inside infected cells or cancer cells. And it is mediated by mainly cytotoxic T lymphocytes. As you know, the pathogen's antigens are expressed on the cell surface of an antigen-presenting cell. The T helper will release cytokines and interleukins that will help activated TC cells to bind to the infected cell's MHC antigen complex and to differentiate into cytotoxic T cells that will lyse the infected or the cancer cells. All these mechanisms will be discussed later. So, a small recap. We have the adaptive specific immunity comprising two major immune responses, the humoral specific immune response triggered by the secretion of antibodies and the cell-mediated specific immune response whose effectors are the cytotoxic T lymphocytes and the T helper. Thank you.